Okay, girls, I'm going to really quickly run through the test with you. So the first question is in photosynthesis. ADP is an organic molecule found in large quantities in most cells. It's converted into ATP in green plants. The original source of energy for plants is going to be sunlight. Yay! The rate of photosynthesis is not affected by which of the following? All right, we're going to be affected by the amount of oxygen in the cell? No, because that's something it produces, so it's a product. We're going to be affected by the amount of carbon dioxide? Yes. We're going to be affected by temperature? Yes. And we're going to be affected by light intensity. So this one says radioactive oxygen is introduced into the environment of an experimental plant. After a short period of exposure to sunlight, the radioactive oxygen is detected in sugars in the leaf. So we've got oxygen that is introduced into the environment, and now we're finding it in the sugars. So what was the molecule that likely introduced it? So was it water? So think about what water does. Water goes through the light-dependent reaction to split to become oxygen. Does it become sugar? No. So it won't be water. It won't be ATP, and it's not going to be ozone. So I'm guessing you were tossing up between water and carbon dioxide. But the correct answer is going to be carbon dioxide. And that's because the oxygen that was in the carbon dioxide, I know it's a bit tricky, but it goes in and then becomes the oxygen that's in glucose, because the oxygen that's in the water has already become oxygen in the air. So what molecule produced in the light reactions is then used in the light independent reactions? There are actually two, but there's only one on your list. So the two that are used are the NADPH and ATP. So the only one you had there was ATP. The O2 is also produced in the light-dependent reaction, but it's not used in the light-independent reaction. All right, which one is evidence for the bacterial origin of chloroplasts? You should hopefully have said the membranes are similar to the cyanobacteria. The cell walls, no, we don't talk about cell walls. The DNA of some chloroplasts is arranged as a linear chromosome. No, it's not linear. It is circular. And plant cells have a large vacuole. Yes, they do, but that's got nothing to do with the fact that they were bacterial. All right, now we have to identify from the diagram above. So what we should have had, oh goodness, the diagram's not going to show right now. But what we should see here is that B was showing the arrow going into the granum. All right, so B should have been what goes into the granum, and that would be water. What is F? F was the product, one of the products that left the light-dependent reaction and went into the light-independent reaction, so that should be water or ATP. And then H was what is the name of this second part, so it's Calvin cycle, and I'm really sorry that we wrote down light-dependent reaction. That's wrong. It should be Calvin cycle or light-independent reaction. All right, really quickly going through cellular respiration. The energy of the electrons in the electron transport chain are going to make ATP. Now, most cells in the body respire aerobically. However, sometimes we switch to anaerobic. So anaerobic does not involve the mitochondria. We're fine with that. It's not preferable because it's not more energy efficient. It doesn't involve the Krebs cycle, so it doesn't work there. And they cannot be obligate anaerobes, which means that they have to do anaerobic respiration, because that just doesn't make sense for this question. So the presence of oxygen glycolysis is followed by not lactic acid, not ethanol, because we've got oxygen, not electron transport chain, because before that happens, you have to go through the Krebs cycle. All right, this one here, glycolysis, what's our net gain? Hopefully we can say that it is 2 molecules of ATP. Structure labeled Y is your cristae. Cristae is where your ETC happens. So this is where you get the majority of your ATP. What's happening at X? X is the inside the cristae, so that's going to be called your matrix. Cellular respiration, 3 stages. Which one one of which is the Krebs cycle. How many molecules in the Krebs cycle? Everything's always 2 unless you're in the ETC. Nice and easy. Which one is not a factor that limits carbon dioxide? Okay, it limits photosynthesis, but it doesn't limit cellular respiration. And the last one, fermentation in yeast produces ethanol. It does not require lactic acid. That's what's produced in animals. It does not require the presence of oxygen. That's just wrong because we're doing anaerobic respiration, and it doesn't involve the Krebs cycle. So then we went to this question here about why animals would produce anaerobically. So it's a bit hard to see, but this top one here, you needed to say something about a lack of oxygen. Not that that oxygen isn't being produced, but just that there is lack of oxygen, so that's why you switch on to anaerobic. What are you going to produce in animals? You're going to produce lactic acid and 2 ATP. And then why is it less efficient? A lot of you talked about it, just rewording the question and saying that it was less efficient. No, you need to say why. So less efficient means that we make less ATP. So I was being very particular when I wrote the mark scheme, and I said 2 ATP versus 36 or 38. But now I'm happy to just say it produces way less ATP, so therefore it's less efficient.